We have seen the different types of tissues which are present in the plants. We came to know what types of cells are present in them. We came to know their functions. We came to know about their location. Now let us understand the actual location. If you observe a particular stem under the microscope, what you are going to find, that would be one part of study in this lesson. And secondly, we are going to understand the epidermal tissue system in specific arrangement that it is having and in little bit detail. And you might ask that why we are studying epidermis in great detail and why rest of the things are not being studied, because there is something strange about the epidermis that you find different types of cells, and that would be little bit interesting for you. Other types of tissues or other tissue systems, they have the simple arrangement. So because the epidermis is having different arrangements, so we are going to get introduced to that now in this lesson. Now when we talk about plant tissues, we have to see their arrangement. We saw that there was parenchyma, collenchyma, sclerenchyma, xylem and phloem. This was what the tissues were all about, and of course there was meristematic tissue. When I talk about the plant tissues, we see that they are going to be present in any part of the plant. Now it is best to study all types of tissues which would be present in the dicot stem. Okay. Now take a young dicot plant and cut its stem and observe it under the microscope. What you are going to observe is, you know how to make a slide. You are going to use a cover slip, you are going to use a stain so that it is easier for you to observe it. Then the stain could be saffron or it could be the green part that oftenly we use green stain, or we use the red stain. And when we observe it under a light microscope, what we see is that there is a particular set pattern of arrangement of cells. Now what is that set pattern? Firstly, because we are talking about the stem, it would be somewhat like this. Okay. The outermost layer is the epidermis. Okay. And there is a central part that is known as pith. This central part has the ability to get lightly stained because it is having very less cells. There is a lot of intercellular space. The cells are very less concentrated or dense over here. This part basically between the pith and the epidermis, the outermost layer, as I told you, we are going to study it in detail. Outermost layer of cells is known as epidermis, the inner part is the pith, and in between you are going to find all the types of tissues ranging from the meristematic to the xylem, phloem, everything would be there in this region. Now what we are going to study, how we are going to study is, because it appears like this under the microscope, of course there would be cellular organization, you are going to find cells inside it, there would be outer arrangement of cells. We are going to magnify this part for our understanding and observe how it looks under the microscope. Okay. Now we are going to see this part's diagram. This is how the stem would be under the microscope. This part is to be highly magnified and understood so that we get an idea what the plant tissue arrangement is all about. Now this outermost one is the epidermis, because we are dealing with plant. This outermost layer is the epidermis. We are talking about the dicot stem. Okay. There would be some epidermal appendages which would be present. Okay. Now because this is parenchyma, what happens, this is epidermis is parenchymal layer, because this is a stem, we find some sort of cellular appendages that is known as stem hair or the trichomes. You are going to study about these in detail as well, multicellular appendages known as stem hair. Now after this there is a layer of hypodermis, because this is a dicot stem. This hypodermis is collenchymatous cells. Now you remember they had a huge central vacuole and hemicellulose depositions at the end. The cells were somewhat hexagonal. That was also one particular condition. So we find collenchyma below the epidermis that forms the hypodermis. This is what is collenchyma. Okay. Below collenchyma we are going to find layers of parenchymatous cells, that isodiametric, lots of intercellular spaces. These parenchymatous cells are typically the cells that we talked about, and they form the cortex. So it would extend till here. This would be all cells would be present over here. Then comes a layer known as endodermis. Closely packed barrel shaped cells are present and it forms the endodermis. Endodermis is the internal layer and it is known as, it is a type of dermis only, but it is internal to the epidermis. Outermost was epidermis, then we have endodermis. I am going to label them. This is epidermis. Okay. This one where hemicellulose depositions are there, cellular angular thickenings are present. This is collenchyma. I am magnifying this thing only, but like this. This part is the cortex that would be made up of parenchymatous cells. I have not made it like this. These are the parenchymatous cells which form the cortex over here. They would take up the strain because they are living. Then we have the endodermis. Now inside the endodermis, this was all simple permanent tissue that you were seeing. Inside the endodermis, you would find the meristem as well as the complex permanent tissue. Now endodermis, beneath endodermis, there are pericycle cells. Again, they are parenchymatous. Pericycle is present, 1 to 2 layer thick pericycle. Okay, like this. Okay. Now beneath this pericycle lies the phloem. Now such is the arrangement of phloem that here you are going to find patches of phloem, not completely phloem, but patches of phloem. There would lie a vascular cambium between this phloem and the next type of cell, that is, or tissue to be very specific, that is xylem. There is rows of xylem which are present, and in between, of course, in order to provide bulk, there is lots and lots of parenchyma. This is followed by pith. Pith is present. Okay, we have reached till that part. Rest of it will have lots of parenchymatous cells in between. Just like this, you can represent it. So this is what you are going to observe under a slide when you study this particular part. Endodermis was followed by pericycle. Beneath the pericycle lied phloem. That was the complex permanent tissue, and this is primary phloem. Then we have the vascular cambium. You remember, it was the secondary meristem. Then lies the primary xylem, followed by the pith. This is what a dicot stem has inside it if we observe it under the microscope. Okay, it looks quite baffling and good to see what the tissue arrangement or the cell arrangement is inside the root. If you ever get to see it under microscope, you are going to enjoy it. So good luck doing that. This is what you have to understand for plant tissue arrangement inside a dicot stem, and if you are performing it, make sure you take a very tender stem, because it is easy for getting a good slide, and it is better to put it in a potato so that the cutting is easier and you get a very thin section. This was the transverse section. More thin, better the view. Okay. Now last thing is that you have to move from the stem to a leaf. Okay. Now when you move to a leaf, what you have to observe is that you have to observe its lower epidermis. Now we know that the outermost layer is the epidermis. In the leaf, if you observe that epidermis, you cut its transverse section, you observe that leaf under the microscope, the lower surface would have abundant arrangement of stomata. Now we are talking about epidermis tissue, epidermal tissue to be very specific. Okay. Now this epidermal tissue is going to be present in different forms. When I talk about roots, because the purpose of roots is absorption of water and minerals, these roots have epidermal tissue modification or arrangement in the form of root hairs. Okay. Now supposedly this is the epidermis, like this is the epidermis, and one of the cells would be highly elongated, its cell wall would be highly elongated so that it becomes thin and it increases the surface area for absorption of water. This is root hair. As we talked in the case of dicot stem, there were multicellular stem hair which were known as trichomes. Okay. In the case of stem hair, we have multicellular trichomes that are usually present for some or other sort of defense mechanism. They do not have absorptive purpose, so they can be multicellular. These have to be unicellular so that they become very thin and increase the surface area. Trichomes are present. In case of leaves, we have stomata. Okay. Now when we talk about stomata, we have to observe the leaf under the microscope. Now what you observe in the lower surface of the leaf is, now supposedly these are the parenchymatous cells, like this they are arranged. In between, what you are going to find, epidermal cells, you are going to find a bean shaped arrangement known as stomata. They are embedded in such a way that there is arrangement of cells in such a way that you find these stomata, so many stomata present. The pores could be open or they could be closed. There are cells present inside them. This is what a stomata looks like, and this would be the parenchymatous cell arrangement of the epidermis. Stomata would be present like this. In this case, we are going to find another stomata embedded. These are nothing but the epidermal cell modifications only which are present. Okay, and they are surrounded by particular type of cell known as subsidiary cells. So these are the openings. Now what you see over here is that this kidney shaped cell is, this entire arrangement is known as stomata. Okay, singular is known as stoma. This is stomatal aperture, opening. Now what is the purpose of stomatal aperture? And of course it is having its guard cells. Each of the kidney shaped cell is known as guard cells. This is to be remembered. What is the purpose of this stomatal arrangement in the epidermis of leaf? The arrangement is responsible for exchange of gases. Now you know that leaf is the site where the photosynthesis takes place. How does photosynthesis take place? It takes place when the plant or the cells of the plant are provided with carbon dioxide. Now from where this carbon dioxide comes? It comes from the atmosphere and it gives out oxygen. Now if there is no arrangement inside the leaf made in such a way so that there is a connection between the outer atmosphere and the atmosphere present inside the leaf, there will not be exchange of gases that could take place. So in order to make that exchange of gases possible through the leaf, you must have seen this. Oftenly what we do is that we make a leaf and through it we say that O2 is coming out while carbon dioxide is going inside, and that is giving rise to photosynthesis process, and this process will give us food, or we say that this will give us glucose. This is the simplest thing that we have understood till now. If not, you are going to study it, that the leaf is going to take up carbon dioxide, it is going to give out oxygen. This carbon dioxide would be used to form glucose. How does this gaseous exchange take place? It has to take place from the leaf surface, and it is these pores surrounded by guard cells which are responsible for that gaseous exchange. Okay, so this was about the leaves and their epidermal tissue. And before we end the plant tissues, last thing is that oftenly this epidermal tissue system that is present in the stem is responsible for providing strength to the plant, and that we see in the case of cork. Okay. Now what is cork? You must have seen the bark of the trees, the tree trunks, they are often very hard. How they are hard? There is an impervious material that is impervious to water made up of suberin. Okay. What you see in this particular arrangement is that the outer epidermis, we have seen the dicot stems section right now. In that you saw that there was collenchyma, there was cortex, the epidermal tissue, some part of it becomes that outer covering tissue, some part of it becomes meristematic, or you can say the part of cortex becomes meristematic all over again. Okay, it was not meristematic in the beginning, it becomes meristematic and it starts giving rise to the cells which have suberin inside them. This suberin is impervious to water, it is impervious to gases. Now when something is impervious to gases and water, it is going to provide some sort of insulation, and that insulation is responsible for giving strength. So in the end what we get to know is that there are many systems in the plant, broadly three: the ground tissue system that has cortex inside it, the epidermal tissue system, and the vascular tissues which had xylem and phloem. We didn't talk about in depth about those. What we talked about is epidermal tissues. We talked about the epidermal tissues in the sleeves which are responsible for gaseous exchange. Because this is the outer covering tissue, it has to provide strength and flexibility. So we talked about the cork and the suberin material, that is the cork that is formed, the bark part of the tree we talked about. Apart from that, we did see that in the epidermis we have root hair for absorption, we have trichomes for strength and stability. So this is all about the epidermal tissue, and with this we come to the end of plant tissue.